The first presenter will be Kara Tracewell from Caltech, and she's going to tell us about the direct evolution of new bacterial communication lines. Right, Kara.
Thank you. So this is a summary of some of my postdoctoral work while I've been in Frances Arnold's lab at Caltech. And what we've been working on are the transcriptional activator LuxR. Okay. So we're all here at the Synthetic Biology Conference, and so something that we're really all interested in is doing forward engineering in organisms. So we want to be able to take apart the organism from its components and understand the interactions of those components and the networks that they comprise and the functions that they give rise to, and then be able to forward engineer those into new genetic circuits that would give rise to, primarily what we're interested in is trying to understand design principles of evolutionary networks. But what the great thing about synthetic biology is you can also actually have applications with this technology. You can create products that are of interest to pharmaceutical applications, doing gene therapy, cell-based sensors, and then all the many other things we've heard about this weekend. So for us, the thing that we've been most interested in is the types of, the organism, I guess you could say we're interested in, is microorganisms and the types of consortia that exist in nature. So these are very interesting because they can conduct very complex functions, so such as degrading cellulose in a biofilm, or degrading a sludge, or using a consortium to produce methane from waste. There's also the many different bacterial consortia that live within our body, which we've heard a little bit about this weekend too, such as those that aid in human digestion, and those also that live as biofilms on our teeth and that actually form plaques. The other interesting thing about consortia is that they many times seem to be better at doing pathogenic functions when they work together in consortia. So if we can understand consortia, how they work, maybe we can have a little bit better understanding of how to prevent those things from occurring and prevent pathogenesis or virulence. So all of these types of microbial communities depend on cell-cell communication. So that's where we've started out with trying to understand better cell-cell communication. And if you can understand that, you can engineer many different things. You can engineer population-level behavior with microorganisms, and potentially you could make synthetic multicellular systems or synthetic ecosystems. The actual communication proteins that we've been studying are those from Vibrio fischeri, and Vibrio fischeri, for those who don't already know, is the microorganism that lives in Hawaiian bobtail squid. It produces light when there's a accumulation of the signaling molecule. So the signaling molecule is made by LuxI. It's a synthase, and these produce the AHL, and it's detected by LuxR, and then in the organism, that turns on luciferase, which causes the bacteria to glow when they're living inside of the squid. Oh, and here's my picture of doing that. So actually, LuxR, the actual mechanism that's been worked out over many years of biochemistry by Pete Greenberg's lab and many others, is that most likely there's two monomeric LuxRs. They're in some conformation that prevents it from interacting with DNA, and it can't activate transcription at that state. It somehow binds these two signaling molecules and forms a dimer, and that dimer then can bind to DNA and recruit polymerase, which results in the transcription translation of whatever gene is following it. So we're interested in trying to engineer a set of, a whole series of proteins that can be activated by different cinders, different signal molecules, and bind through different DNA sequences. And in order to do this, we don't want there to be any crosstalk between them. So we don't want another sender to be able to activate at this particular DNA sequence with a different sending molecule. And vice versa, we don't want a molecule of LuxR that can be activated by something made by LuxI. We don't want anything by LuxI to also activate a different molecule. So we want to be able to try to engineer two orthogonal pairs, which would be great. So the limitation, though, is in nature, all these different LuxR homologs that are known in microorganisms, they generally all activate at a very similar DNA sequence. By similar, I highlight in red, everybody has basically a CTG at the 3, 4, 5 positions. There are a little bit of variation to that, but it's not very much. And we've gone through and done a lot of mutagenesis with the LuxBox and found that these are actually the key base pairs in recognition. So it seems the whole family is very tuned to that particular sequence. So if we want to have 2 orthogonal LuxRs, we'll have to engineer the DNA specificity of the protein. So also in nature, if we compare the different signal molecules that are recognized by all these different LuxR homologs, we notice that there's a few general themes. They all have this acyl homostyrene lactone, and they all have this first oxo group. But sometimes they have an oxo group at the one position, and sometimes they don't have the oxo group, and sometimes they do. And LuxR happens to recognize 3OC6 that does have an oxo group. And that's made by the cognate AHL synthase LuxI. LuxI also makes a little bit of this molecule, which doesn't have the oxo group. So 2, LuxR homologs that we do have a little bit of structural information of are these 2 here that I highlighted. This is LASR, and it recognizes 3OC12, so it's a longer acyl chain, and TRAR, which recognizes a C8 acyl chain. However, these aren't very useful as far as being able to design in a new property. In my opinion, mainly TRAR is only 20% identical to LuxR, and LASR is only 40% identical. So it's not a lot of sequence homology to use for making any kind of structural comparisons. But it would be nice to make a standardized part list from these. The problem, though, with all these other homologs is some of them activate as dimers, and other ones activate as by pairing together into a pair of 4 in order to activate. And they all interact with different RNA polymerases. So, and not all of them activate in LuxR, in E. coli, so they don't all function. So it'd be nice, our idea is basically just to take LuxR and engineer it to do all these other functions. And so to begin with, doing directed evolution, we generated a screen. And in this screen, basically, we have LuxR is turned on constitutively, and it responds to a signal that we add. And then that activates the LuxI promoter and turns on GFP. And so you can just do a screen and look for fluorescence to look for activity. And this was done, and we could find that we could generate LuxR variants that respond to something that has no oxo group and a longer acyl chain, so C10 homostyrene lactone. And that's what I show here. G2E is that variant. But G2E can also respond to 3OC6. And so it can actually respond to quite a few other things, too. And that's a commonly observed thing when you're trying to do in vitro evolution. You find that you just end up broadening the specificity of the protein that you're trying to do evolution on. Another way to try to do this in a more high throughput fashion is instead of doing a screen, actually doing a selection. So you're trying to only look for something that can turn on the chloramphenicol resistance gene and provide survival for the bacteria. So we set up a selection system that could do this, and we called this on selection if we can select for that activity. But to overcome the previous problem I described of having broader specificity, we set up also something called off selection. So in this case, if we actually do turn on the gene, if we turn it on here, we're turning on BLIP, which is beta-lactamase inhibitory protein, and the cells contain beta-lactamase. So this protein will actually turn off beta-lactamase, and it makes the cells carbonicillin resistant. So if something's acting at that promoter, it's killing the cells. So now all we have to do is take advantage of this, design in something that we don't want the LuxRs to have that property anymore, and we can select for things that don't have that. And in general, using this system, the beta-lactamase inhibitory protein for the off selection and anything for the on selection is a nice system for any type of on-off circuit that you want to try to create with synthetic circuits. So just to reemphasize, the nice thing about doing these selections is it's a lot easier to pick out the yellow tulip out of the whole field if you're doing a selection, because you can look at quite a few more things. So you can, more than likely, you can find your yellow tulip. But the big difference between doing the screen is you don't have the screen and the selection. For the screen, you have to go through and actually look at each tulip and say, is it red or is it yellow? But for the selection, what we do, we just plate the cells. We come back the next morning, and all of the red tulips have died, and only the yellow tulip is alive. So it's a lot easier to go through quite a few more variants of proteins. And so we did this, and we were able to find a variant then called G2E R67M. It can activate at C10, and it doesn't activate at 3OC6 anymore. So it's changed its specificity of the protein. And just briefly, that mutation in TRAR, the structure that we also have a structure of that protein, it's actually located towards the end of the acyl chain, so it doesn't make a lot of sense why that mutation would work. But if you look in the structure of LASAR, that mutation is about 5 angstroms away from the second oxo group, so it changes the hydrogen bonding interaction and thus changes the binding of that acyl homoserine lactone. But there's a problem because LuxI also generates C6, and that variant that we found can still activate at C6. So we had to then do further evolution where we did off selection with C6 present, and we could find actually quite a few solutions to that particular problem that have at least 3 amino acids that change the specificity so that it's still activating now at C10, but it's got drastically reduced activity towards C6. And it retained or got better at not responding to 3OC6. And that series of mutations is found basically in that beta sheet, but they're all positioned sort of away from the actual acyl homoserine lactone. So it's not clear actually how they change the specificity of the protein, other than there's repacking of the protein around the small molecule. But the one other thing that in general, all the amino acids were being changed to more hydrophobic side groups, and that makes sense because it's trying to recognize something now that's a longer acyl homoserine lactone. We also did evolution then and found a variant that could not respond to C10. So what we did find along the course of all this work was that we could get LuxAR actually to activate towards C10 if we let the cells grow for a really long time or to a really high density in our assay. And so that could be a problem if you're trying to plate these on trying to plate the cells and create patterns, because after a while, you're going to start to get activity towards C10. So we did on-off selection and found a variant that has 3 mutations that, again, are found in that beta sheet for those 2. This one's actually on the surface. But they don't seem to directly interact with the 3OC6 or the AHL. We also applied the same on-off selection to changing the specificity of the DNA binding domain. And that's what I show here. It's just an inverted repeat. And we went through and made all of the symmetric mutations of the LuxBox and looked for any activity on that to find the ones that would be good to do directed evolution. And the ones that are the most important, I circle here, where if you change those to anything else, you're not going to get any activity. And what we could find was that when we changed it to the G position at 5 to A, we could find a number of variants that activated that. And our best, I would say, would be that the best change in specificity is we actually have activation here better at G5A than we have at the wild type sequence, whereas LuxAR wild type is not activating at all at G5A. We took all those variants and tried to take them further to do evolution to get them to either respond better, have better specificity for G5A, or to then add additional mutations to the LuxBox. but we were unsuccessful in any of those attempts. But that position, 212, that seems to be pretty important. If we look in the only crystal structure we have of a DNA binding domain homolog, it appears to contact the phosphate. So it's also not clear why that would actually be changing the specificity, but it does seem to work. And some previous biochemical work showed that position R212 is probably very important for DNA binding. When Greenberg's lab did alanine mutagenesis, they found that the position 212, when it went to alanine, it didn't work. The other mutation we found at position 232, we weren't sure exactly what that was doing, but after purifying the protein and comparing the melting of the protein by CD spectroscopy, it appears to stabilize the protein substantially, so it can tolerate more mutations. So we also looked at LuxR at different LuxBox, as I mentioned before, and we could find that it activates at 3T and 4C. So we took advantage of that. So now instead of trying to take LuxR and evolve it to something that's completely orthogonal to the LuxBox, we've actually got variants that can respond to G5A, and then LuxR can respond to these LuxBoxes. So we do actually have something that is orthogonal in its DNA specificity. And I just show that here. We have activation of this mutant at G5A, whereas LuxR is not responding, and LuxR is responding to position 3T, whereas the variant we found is not. And we also found if we used a stabilized protein, we could get actually better activity at that position and also here. So we want to take advantage of the actual modular appearance of LuxR. That is, there's an AHL binding domain and a DNA binding domain. The TRA-R structure, there's very clearly a linker between the two domains. And so our idea was simply, can we just swap around the domains and maintain the specificity that we'd evolved previously? And so we're combining that then with the two domains that we found previously, the G2FD3 that responds to 3OC6 and not long acyl chains, and this variant, our best variant, C10C, that responds to C10 and not to 3OC6 or C6. And that is, in fact, what we find, that we can get activity with the long acyl chain at that new LuxBox. However, one thing we did find was that if we expressed both variants in the same cell, that they maintain their specificity. They activate as they did before in response to their respective signaling molecule at their respective DNA sequence. But just having the other variant expressed in the same cell reduces the activity, which is a little bit of a surprise because we were under the impression from all the biochemical work that they actually don't dimerize until they bind AHL. But it appears that they can form dimers prior to binding AHL, and that when they do, they form inactive heterodimers. So we would like to be able to select for proteins that act as homodimers, and we've set up the selection system to do that, and all I can say at this point is that's in progress. But the variants we have found aren't completely useless. What we've been able to demonstrate is that our variants, when we mix them with respective AHL synthases, that they have orthogonal AHL specificity. So here is a plate where I took LuxI and put this in one cell strain, plated it in top agar, and then dripped onto it a drop of cells that contain the the LuxR variant and the plasmid for expressing GFP. And we see we have activation of that variant that we found earlier, and we also have activation of our new variant that doesn't respond to C6, and also LuxR. And then here, if we do this with BTI2, which is an AHL synthase from Burkholderia thaliadensis, it makes C10, and all of our variants respond to that, and they don't respond to anything produced by LuxI. You also notice that all of the BTI2 variants are a lot brighter. Apparently BTI2 works a lot better than LuxI does. It makes much more AHL in our E. coli cells. And I also show here our variants, our recombined variants that activate at G5A, and they activate just as well as the wild type does. Or sorry, as that variant does at the regular wild type Luxbox. So we were able to then take these LuxR variants and combine them in a microbial consensus circuit. All we've done is take one variant and the other synthase and put that in one cell strain, and then the other pair in another cell strain. So now one strain can turn on the other and vice versa. And if I streak them out next to one another, maybe you can't see that quite here, there's GFP forming at this interface by these cells, and there's mCherry being formed by cells at this interface. But you probably are wondering why they look like they're pretty well matched. They're actually, well, they're not that well matched, but we did have to weaken the ribosome binding site for the BTAI2. We also had to add an LVA tag, and we had to add an LVA tag to increase protein degradation of the mCherry. And we just about have a system that's matched again. So in conclusion, what we've shown is that directed evolution using dual positive and negative selection is a good way to generate new parts. It's also a great way to improve the properties of existing parts. And we were able to identify variants of the DNA binding domain and those that are orthogonal in their AHL binding specificity. And those variants could then be recombined to generate activators that have properties of both variants. But the general thing to conclude with all directed evolution is just be careful how you set up your screen or your selection, because that's what you get, whatever you've selected for. And just to conclude my acknowledgments, to Frances Arnold for all of her great support, and Cynthia and Lutz for their contributions to this work, and also to Ricky Ulrich for the genomic DNA for the BTAI2. And thank you for your attention.
Thanks, Kara. So the floor is open. Any question? Yes.
I was just wondering, especially with your on selection, did you ever run into issues where the that Lux promoter was being mutated, so it was no longer being repressed effectively, and that's why the cells were surviving on chloramphenicol?
Well, so what we do, we go through multiple on-off rounds to make sure we're amplifying something that's working, and then we reclone it. We reclone just the LuxR, put it back either in the selection system or do a screen with it. So yeah, we do have mutants that crop up all the time, but by going through multiple on-off steps and by recloning it, we identify variants that way.
Was it common, or was it fairly rare that might happen?
It's more common with the off selection to find variants, because you're putting in there beta-lactamase inhibitory protein, and the Lux promoter is actually a little bit leaky, which probably a lot of people who've worked with it have found. And so it's leaking out a little bit of the blip, and the cells don't really like blip. So they just try to mutate and get rid of it. So we can't grow the cells for much longer than 14 or 15 hours. After that, I don't really trust that what I've found is really a LuxR variant.
If you have one more question.
Sorry, just along the same line, regarding that counter selection, what would the advantage of using blip versus using something like SACB or a different counter selection?
There's probably not an advantage. It's simply an off selection. Yeah, I think any off selection is useful for that. Yeah.
All right. Thank you, Karen.